Hi, in this video you are going to learn about the transport in phloem. In the previous videos you have learned about the transport in xylem. So now let's see how the transport in phloem happens. So let's start with learning the structure of phloem vessels. All right. So there are two types of types of cells in the phloem tissue. The first one is called sieve tubes, and the other one is companion cells. So sieve tube are this bigger tube, all right, and while the companion cells is this one. Okay, one for the lower tube and the other one for the upper tube. So as you can guess, it has nucleus in the companion cell. So cells of phloem are actually a living cell. They are still alive. Even though a sieve tube seems to be empty, but actually it keeps its cell membrane, and because of it doesn't have nucleus or ribosomes, they don't have to worry because companion cells still have it, and actually they can communicate, and these companion cells can provide uh, the protein or anything that is needed by the sieve tube because of lacking of nucleus, right? So both types of cells are alive, and sieve tube cell wall that are connected to the top and the bottom of the cells, as you can see from here, the end of the sieve tubes are perforated, or it means that it has so many pores. As you may guess, it is to accommodate the movement of the liquid sap, or the sap of the phloem, the liquid inside the phloem, to pass through. However, as I said, it lacks of nucleus and ribosome, and the main function of this cell is to transport sugar. Now, each sieve tube is accompanied by a companion cells, like what I said before. This for the upper one, for the lower one, and they still have nucleus and ribosome to make protein needed by the sieve tube. So they have to cooperate together. So then maybe you ask, like, how they can transport the protein that is produced by the companion cells to the sieve tube. So basically the sieve tubes and companion cells have holes that make them connected, called plasmodesmata. And the definition of plasmodesmata is open channel in the cell wall which cytoplasm of both cells are connected. So the companion cell will produce protein needed by the sieve tubes, while the sieve tubes remain empty, even though it's still alive and has cell membrane that can do active transport. It has to be empty because it will accommodate the movement of the liquid inside it. So in this slide, I will give you a brief explanation of what phloem cells can do. But in the next slides, we will talk about the processes in more detail as step by step. All right, so let me remind you once again that phloem cells are alive. So they can do both passive transport and also active transport. Now, active transport is needed. Why? Because they have to take as much sugar as possible from sugar producing cells. And this kind of cells that produce sugar is called sugar source. Now, for example, in this picture, you can see leaves. The leaves has palisade mesophyll as the main photosynthetic site. Because of that, they must have lots of sugar. And phloem will do active transport to take most of the sugar. Alright, next, when sugar is accumulated inside the phloem cells, the high concentration inside the phloem cell will attract water to do osmosis to enter the cell of the phloem and create water pressure to go to places that need sugar. And the cells that need sugar is called sugar sink. So you can see in this picture, phloem and xylem, as you have learned in the first video of this topic, are placed side by side. So the high concentration of sugar inside the phloem will attract the water from the xylem to enter the phloem tube, sieve tube. And then this will create high pressure of water because more and more water is added, and then they will flow to the place where the sugar is needed. Okay, and the cells that needs the sugar is called sink cell or sugar sink. However, I have to remind you before we go further to the details of phloem mechanism in transporting the product of photosynthesis, I have to remind you that sugar source is not always leaves. Yes, most of the time it is the leaves. Okay. Now, for example, like during germination, leaves are actually the sink. So, for example, this is the seed that has lots of product of photosynthesis from the parent plant, right, that is stored in the endosperm of the seeds. So when the seed is germinating and creating the first leaves, right, so actually these leaves are not yet sugar source. They still need the food from the seed or the tuber to create bigger and bigger leaves. All right, so it means that in this case leaves are the sugar sink and this tuber is sugar source. However, in other conditions, during growth and reproduction period, leaves are the source of sugar. So for example, in this case, leaves that produce are quite a lot and they can now do photosynthesis very, very well. Now during reproduction time and during growth, they'll start to produce sugar that can be stored in the storage organ. So for example, in this case again is the beetroot. Okay, now so the condition is now reversed. The leaves is now the sugar source and the tuber is now the sugar sink. Alright, if you have understood the difference between sugar source and sugar sinks as well as the outline of the phloem transport, now it means that you are ready to learn the phloem transport step by step. Alright, so for this time, let's just assume this is summertime or during growth stage, so leaves are the sugar source. Alright, let's talk about step 1. You can see step 1 over here. This is step 1. Alright, now step 1. Sugar is taken by phloem cells using active transport from the sugar source. Okay, so this is the line. Next, step 2. Which one is step 2? This one is step 2. Okay, step 2. So step 2. High sugar concentration in the phloem attracts water from adjacent xylem vessel to enter the phloem by osmosis. So this line shows that the water attracted to enter the phloem due to the concentration gradient. Alright, and this happens through the process of osmosis. This creates high water pressure inside the phloem. Again, your syllabus wants you to remember a specific name or terminology for this high water pressure created in the phloem. The name is hydrostatic pressure. Now, this pressure has a force to go to any direction that has lower hydrostatic pressure. Of course, the one that has lower hydrostatic pressure is the sink, because as you can see in this area, the phloem surround the sink cells. They have a very low concentration of sugar. Of course, water will not be attracted to enter this phloem, therefore there is no pressure of water will be created. The water pressure will come from the place where the sugar has been uploaded by phloem from the source cells. Alright, let's continue. Thus, unlike water in the xylem that always moves upward, liquid in the phloem can go anywhere. So, don't get it wrong, it is not always go to the roots. If there is a fruit, for example, this is a plant, okay, and there is a branch, on the top of it there is a flower or fruits that is growing, it means that the water pressure also can go to that direction. So it can go to the left, to the right, up and down. That's the nature of the direction of phloem movement in the plant. So now the water with the sugar now flow to the direction of the sink cell or the sugar sink. In step 3, sink cells actively take up the sugar from the phloem sap. Now the sugar is actively taken by the sugar sink. Now here comes the last step. Because of the take-up of the sugar from the phloem by the sink cells, the concentration of sugar inside the phloem now decreases. Due to lower concentration of sugar compared to mineral in the xylem, right? Water will leave phloem and re-enter the xylem. You can see the arrow over here. So it will re-enter the xylem. So basically water in the xylem are just acting as the current to bring the sugar into the sink. Okay, and then this causes the hydrostatic pressure in the phloem near the sink to remain low. Again, the pressure become low because the water leave the phloem. Thus, this will ensure continuous flow of sugar from the source of the sugar, right? So you can see that if the water comes back here, right, and then again because of the transpiration pool they go up again, and then in the leaves the phloem take up the sugar again, and then the water will again enter the phloem by osmosis, flow to the sink take the sugar, the water leave and go back to the xylem, and so on and so forth. So it will create a cycle. I hope you understand the details of the phloem transport, okay, by the plant. Now you have learned the details step by step of how phloem take sugar from sugar source and have water from the xylem to transport it to the sugar sink. Now, however, your syllabus wants you to learn more detail in cellular level, okay, about how the active transport of the sugar happens in the membrane of the sieve tubes, right? Shall we start? The first point that you have to know, the main sugar carried by the phloem sieve tubes is sucrose, alright? So, the representative of the sucrose is this triangle with the letter S. This is the sucrose. Now, for your information, this area is the inside of the sieve tubes and this one is the outside of the sieve tubes. So, the first step that will happen is, inside the sieve tubes, there are lots of proton in form of the hydrogen. Alright? Now, if you still remember the electron transport chain, there is a proton pump. So the similar thing that happens here is that the proton, using the ATP from the sieve tube, it will be pumped out to the outside area. Alright, so what happened next is, it's kind of similar with the cell respiration. Yes, there will be accumulation of hydrogen or proton, and this accumulation has or stores some energy, the potential energy. So what will happen next is step 2, which is similar, kind like ATP synthase. The proton will go back inside to the sieve tubes. And this will really, as this happens, it will release some energy. And the energy will be used to take the sucrose, go inside the sieve tubes. So let me read the step 2. Another protein called the co-transporter protein. So let me clarify that this type of protein is called the co-transporter protein. Co-transporter protein, right? So it will allow the proton to diffuse back, and the energy released by the proton will be used to move the sucrose into the cell. Companion cells also use this co-transporter mechanism. Do you still remember companion cells? Yes, companion cells are the cell that is located next to the sieve tube, and it still has nucleus and ribosome. Now, to speed up this sugar or sucrose take up, the companion cell also help the sieve tube to take the sucrose by using this co-transporter mechanism. However, after they have the sugar or the sucrose inside the phloem, inside the companion cells, this sucrose will be given to the sieve tubes through plasmodesmata. Alright, I hope you understand the cellular mechanism of how the active transport of sucrose happens in the sieve tube. There is an experiment that you have to learn based on the syllabus about how the rate or the speed of transport of liquid in the phloem can be measured. Now, however, maybe you will have a question regarding the ethical issues in this experiment. Why? Okay, let me explain about it. So in this experiment, to calculate or to observe the speed of flow inside the phloem, the scientists use a certain types of insect species called aphid. Now this aphid, if you see, it has a mouthpiece. Under the microscope, you can see that the mouthpiece can penetrate through the stem and reach to the phloem. Of course, to take the sugar, because they are basically the insect that we can consider or farmers can consider as pest. Alright? So they can penetrate to the phloem and take the sap containing sugar in it. In this experiment, the scientists will let some aphids to inject their mouthpiece. By the way, the mouthpiece is called stylet. Alright? Now, after their stylet is injected through the stem and reach the phloem, sadly but true, they will be cut or separated. The head and the body will be separated from the stylet, like in picture E. So the stylet will remain, right, penetrate, okay, while the head and the body of the aphids will be gone. However, the sap from the phloem will drip out. Why? Because if you still remember, phloem has hydrostatic pressure, and this water pressure will cause some of the phloem sap to appear at the remaining part of the stylet. Alright? This is the details of step-by-step of method of working for this experiment. Now please take a look at to this diagram. Step 1. 2 colonies or aphids are introduced to 2 different parts of the plant with a certain distance apart. So you can see from here, 1 colony, 2 colonies, and they are separated as much as 65 centimeters apart from each other. After that, the scientists will let the aphids to inject their stylet up to the phloem, okay, to reach the phloem in the stem, and then sadly but true, they will be cut off. Alright, so the stylet will remain while the head and the body of the aphids will be gone. Okay, and then step 2. The plants will be given radioactively labeled carbon dioxide. So using this glass chamber, the plant will be exposed to carbon dioxide that has been labeled with radioactive. Can you guess what is the function? Yes, for the carbon dioxide, it will be used by photosynthesis so that the sugar that will be produced also contain radioactive. Okay, the sugar will also have radioactive. And then let's do step 3. Let's assume this leaves has done photosynthesis process using the radioactive carbon dioxide, so that now the phloem in this part are ready or actively take out the sucrose from the mesophyll of the leaves, and this sucrose contain radioactive. The phloem sap will then go through the phloem, and remember, as they go, the stylet of the aphids will have drop of the phloem sap that contain radioactive sugar. This phloem sap will be collected using revolving plate in a certain time interval. So this plate, as you can see from here, there are lots of holes in it to collect the phloem sap. From time to time, with certain interval, they will revolve, so that the scientists know at what time that the phloem sap that drips out contain the radioactive. The emergence of radioactive phloem of sap of both located is then observed, right? And then the last step. After they got it, for example, in the 50th minute, the drop of the phloem in this area contain radioactive. and then after, let's say, 100 minutes, the first drop of phloem sap that contain radioactive is found in this spot. So it means that within 50 minutes, okay, the phloem sap has traveled as much as 65 centimeter. So you can see that the speed or the rate of flow in the phloem in this case is 65 centimeter per 50 minutes. All right? Or you can simplify, let's say it is about 1.3 centimeter per minute. Let's see. Okay, so that's how what we do in step 4, which is the difference in time of emergence of the first phloem sap with radioactive compared to the second place will be divided with the distance to get the rate of the transport. Now, I hope you understand this experiment. However, again, like what I said in the previous slide, it's an unethical experiment because we know that based on the IB rule, we cannot harm any organism, no matter how small or how insignificant they are. So it is a challenge for you in the future, or maybe future scientists, to think about a better method. Instead of using the APIT stylus, they can create a micro pipette or something that will also give impact, which is the phloem sap can drip out from the phloem. Alright, I think that's it for the phloem transport. I hope you understand the whole concept. Don't forget to also read your textbook. Alright, thank you.